This talk, just given the introduction into microbial communities, I'm not sure how much it has to do with microbial communities directly. It is an experiment that looks at the evolution of multicellularity. It does use microbes as experimental tools. But I would say, although it seems a bit of a long shot, actually multicellular organisms could be thought of as one perhaps extreme manifestation or one extreme consequence of selection working on populations of microbes. And I say that as one extreme. There's a gradation. The extent to which selection actually sees microbial populations is a question that I think is unresolved, and it's one that we haven't really begun to think about too much. Here is an extreme example of how selection gets to see communities of microbes to the point where they come effectively to function as a multicellular organism. Life, it appears, is hierarchically organized. We see that by simple observation. We see that there is a capacity for reproduction that exists at different levels of biological organization. So take us, for example. Here we are here, or we may like to already descend down one. We have a capacity to reproduce as members of a species. We are, of course, in turn made up of cells that have some autonomous capacity to reproduce. Cells include nuclei. They also include organelles, which have an autonomous capacity to replicate below that of the cell. And we could go down further. Chromosomes, genes within chromosomes. So life appears to have a hierarchical structure. Whenever there is a possibility of reproduction, which marks each of these hierarchical levels, there's the possibility of a Darwinian process. It follows then that there is the possibility that selection works at different hierarchical levels. In fact, not just a possibility, it is in fact reality. To me, understanding how this hierarchical structure emerged in the first place is a fascinating problem, and understanding how these lower level units became subsumed and part of higher level units on repeated instances during the evolution of life really does strike to the heart of our understanding of biological complexity. So these Darwinian processes that are possible once we have entities that reproduce are worth clarifying further. So here we have an individual cell. It's a member of a Darwinian population. These are Darwinian individuals, and they are so because of three important properties. First of all, variation among the entities. Secondly, capacity for reproduction. And thirdly, when cells reproduce, they give rise to offspring that resemble the parental type, so there is heredity. So these three properties, the three Darwinian properties, ensure that these entities, cells in this particular instance, participate in the process of evolution by natural selection, and they do it whether they like it or not. By virtue of these properties, these cells participate in the process of evolution by natural selection. And we see that same process possible at the level of individual entities. Here is Stick Girl. She's also a member of a Darwinian population by virtue of manifestation of those same properties of variation, reproduction, and once again heredity. And again, these properties ensure that at the level of these multicellular entities there is a Darwinian process, just as there is a Darwinian process at the level of the cells that make up Stick Girl. Also, by way of background and an essential aspect of this hierarchical consequence, I should say, of this hierarchical structure, is conflict. What is good for the functioning of the higher level is not necessarily good for the functioning of the cells that make up Stick Girl, although Stick Girl, of course, depends on cells functioning for her to remain viable. These cells here must play by very strict rules of development. And that may even involve, and does involve, apoptosis, cells directed to commit suicide as a consequence of developmental cues. This must happen if she is to remain viable. Natural selection, though, works on the short-term interests of entities. Because these entities can participate, the cells, in the process of evolution by natural selection, we would expect to find mutants arising that no longer play by the rules of development. And these cells, should they arise, will reap a short-term advantage relative to these other cells here. If they grow unchecked and unproliferated, of course, this brings about the demise of Stick Girl. This, of course, is an instance of cancer. So cancer itself is a direct consequence of conflicts between levels of organization that themselves are direct consequences of this hierarchical organization of life, with levels nested within levels. My interest here is in the evolution of multicellular life. Here we have a set of individual cells. So once upon a time, early on in life's history, there were just individual cells. There was no multicellular life. These individual cells are each Darwinian individuals and members of Darwinian populations. Something happened, actually on around about 25 different occasions, it's thought, in all three domains of life, that resulted in selection appearing to shift from individual cells to collectives of cells, where the collectives, just as you saw before, are Darwinian in the sense that they manifest variation, reproduction, heredity, heritable variance and fitness. That same capacity for evolution manifests here as well. So how does selection shift during this transition? It's not obvious how it shifts, it's not obvious why it should shift. And depending on how one poses the question, I can put it in particularly problematic or intriguing terms, which are not necessarily the most helpful, I think, in the long term. But one could look at these collectives of cells in a multicellular organism such as ourselves and recognize that the vast majority of cells that make up our body are an evolutionary dead end. It's only the germline that goes on. So you might ask, what trick of nature brought about the foregoing of individual rights to reproduction that was part and parcel of the life of these individual cells, such that they came to replicate as part of a corporate body, where the vast majority of those cells are in fact an evolutionary dead end. But here, though, is, I think, a more useful way to pose the question: how does selection shift? Now, again, selection cannot simply choose to shift. Any capacity to shift depends on those Darwinian properties, heritable variance and fitness. So for selection to shift, there needs to be some sort of handle upon which it can pull. And the primary handle here is the capacity for these earliest groups to reproduce to some extent or the other. So this problem here, how does selection shift, boils down, I think, to asking about the origins of reproduction. Not of individual cells, that's a given at the starting point, but the origins of reproduction of these collectives. This is an evolutionarily derived state, reproduction, and it needs an evolutionary explanation. It doesn't simply come for free, as has often been assumed. I should point out here, too, that although selection shifts in this cartoon, it doesn't stop functioning or acting on the lower level units, just as we saw in the cartoon of Stick Girl and the cells which are in conflict with her functionality. Three stages, then, that I will come back to time and again throughout this talk, in the transition from individual unicellular state to multicellular organisms. The first stage can be thought of or described here. It involves the evolution of groups, typically groups of cells that are cooperating in some way. Individual cells are highly unlikely to remain in close proximity to one another unless there is some benefit for doing so that outweighs the costs of intense competition, swimming around in the waste products of competing cells. So typically the formation of groups involves the formation of some sort of cooperating activity where the collectives of cells reap some benefit that outweighs the costs to individuals. That necessarily sets the scene for conflict, just as we saw earlier on. And the conflict that can emerge here certainly can bring about the demise of this cooperating group. And that's led a number, Rick Michaud in particular, to suggest that the evolution of multicellularity is really about the evolution of mechanisms that police and that punish cheating types. The next phase, though, is what I refer to as the evolution of individuality. So we've gone from a simple group that need not have any capacity to reproduce in its own right to a collective that now has this capacity to reproduce. Once we have this collective, or these collectives with this capacity to reproduce, they are effectively Darwinian in nature. These collectives will participate in the process of evolution by natural selection, such that the next step, the evolution of some form of biological complexity, multicellular complexity, is a done deal once we get to this stage. So I think this is the tough area, how we go from the first simple undifferentiated groups to groups that leave group level offspring. Once this has happened, we have a transition in individuality in this Darwinian sense. So here we have Darwinian individuals with heritable variance and fitness. Here we have Darwinian individuals that manifest heritable variance in fitness. Going from here to here involves the evolution of these Darwinian properties. Let me introduce two super young scientists, Caroline Rose, who's just finished her PhD, and Katrin Hammerschmidt, who's just about to depart on her way back to Germany, having been with us as a postdoc for 5 years. And they have handled tens, if not hundreds of thousands of microcosms during the course of this period in an experiment that is almost inhumanely cruel to contemplate. Just as well I didn't have that sense early on when I encouraged them to get involved in this work. But they've done a fantastic job. Let me take you right back to the beginning and think first of all about the emergence of simple undifferentiated groups, which I didn't say when I introduced you to the cartoon, but this happens readily. It happens in laboratory cultures and many labs around the world. It happens in ponds on the outside. The bacterium that we use for these studies is a Pseudomonas. It's a common saprophytic bacterium. It came originally from the phylosphere, in fact, of sugar beet plants at the University Farm at Oxford. It does nothing particularly special, but it has become a model for much that we do. This bacterium, like many, manifests a smooth colony morphology on an agar plate, and put in a static broth microcosm, it grows in the broth phase. It experiences very strong diversifying selection here and intense competition for oxygen that's depleted thanks to the growth of this type in the broth phase. That establishes conditions that favor, among other types, these wrinkly spreading colonies that produce this mat at the air-liquid interface. This is a simple undifferentiated group, and I'll take you through some of its basic features in a moment. For the rest of the talk, I will use a cartoon to represent the smooth colony, a simple sphere, and a Velcro-studded sphere to represent the wrinkly spreader type, indicating that it's a sticky type in effect. So these sticky types, by virtue of glues, can link themselves one to another. At cell division type, they do not completely separate, and this is the key to forming this mat at the air-liquid interface. These smooth, non-sticky types simply grow in the broth phase as single cells. So the formation of this mat here is an example of a simple undifferentiated group of cells, and the evolution of these in their own right was something that has interested me for a long period of time. A little bit more about the formation of these mats. So here is a microcosm. We fill it with broth and then inoculate it with that ancestral non-sticky type. It grows, of course, and sooner or later we see mutants arising of the wrinkly spreader, the sticky type. And these mutations, of which there are literally hundreds of different single point mutations that can give rise to these types in multiple different loci, I will take you through a few examples in a moment, cause the constitutive expression of enzymes that make a secondary signaling molecule, cyclic di-GMP, and this overactivates enzymes that make glues. In this case, the primary glue is an acetylated cellulose polymer. So single mutations, simple mutations, give rise to this phenotype that colonizes the air-liquid interface. Individual cells that overproduce the glue grow more slowly relative to these cells. They seem to succeed, though, because being part of this collective ensures access to oxygen, which is non-existent in the broth phase. Non-existent because the organisms themselves, through their own metabolism, have decreased and removed oxygen from that broth phase. That mat can grow into a very substantive structure, the cells underneath reducing in numbers because of the fact that they have no longer access to oxygen. We would expect to see, given these circumstances, the emergence of mutant types that would grow in the mat, take advantage of the collective good, which is the production of this mat or the formation of this mat, paying no cost if they do so, and reaping all of the benefit. And we'd see exactly that. You see the emergence of types that are phenotypically similar to the ancestor. Importantly, they are non-sticky, they don't produce the glue, they grow at a faster rate. Held up in this newly created niche, they achieve plenty of oxygen, they grow fast, they pay none of the costs of producing the glue, so they grow even faster, but they contribute nothing to the substance of that mat. And the strength of the mat reduces. That's something that can be measured, and the mat eventually collapses. It collapses prematurely, depending upon the frequencies of these types. So let's go back to this cartoon that you saw a moment ago. The evolution of simple, undifferentiated, cooperating groups from single cells, we've just seen that. We've also seen the demise of these collectives. And of interest, again, for evolutionary transitions is getting to this stage. And again, it's not obvious how these groups go further. They're effectively an evolutionary dead end, like soma. And I've already indicated that it seems that the critical event is some capacity for reproduction. Now typically, and you may wonder why I'm laboring this and don't get to the point more quickly, you would think about this as simply fragmentation. It seems to be a very obvious way by which groups might beget new groups. Indeed, fragmentation is one way forward, and I'll take you through some experiments that look at fragmentation, but it's not the only way. And also there are a good number of reasons, which I won't go into now, but we can talk about later, and perhaps I'll touch upon, why fragmentation is probably not a very good way of reproducing. It carries with it a lot of problems, difficulties in dealing with conflicts that emerge, and it's interesting to look at extant multicellular life. And I know of only one multicellular organism that fragments, a very strange organism called Trichoplax. The rest, irrespective of whether they have a soma-germ distinction, reproduce via a single cell bottleneck, and often via a life cycle that involves multiple stages. And in fact, it is necessary for there to be multiple stages if we're to avoid fragmentation.
What about corals?
What about corals?
They reproduce by fragmentation.
Yes, they do. Yeah, they do. There are some examples that do reproduce by fragmentation. So how they then control conflicts that might emerge is an interesting one. But corals kind of do this by virtue of the fact that they have a structure that prevents the possibility of cells moving around. So I don't think you could get any, I mean, interesting, do you get cheating in corals? I mean, and to what extent is a coral a cooperating?
You get tumors sometimes.
Do you? Yeah.
But you're right, they're rare.
Yeah. A bit like plants, where the structure prevents. Yeah, yeah.
That may be the argument, that's good.
Yeah. Okay, so this is the focus of thinking. Okay, so I'm going to tell you about experiments in a moment where hundreds of thousands of microcosms have been manipulated. Now, the fact that we've done these experiments in microcosms is both a strength and a limitation of the work. A strength in the sense that we keep things discrete and carefully controlled. A limitation because if I just show you and don't present you with this view, you might not see the possibilities for expanding a vision of what we've done to a more general sense. So here's an attempt to do that. And at the same time, to give you a sense of how a life cycle might emerge that doesn't involve fragmentation and maybe from the outset rather more interesting in terms of possibilities presented to selection. So here we have, again, you saw a moment ago, our mat. We'd seen the mat collapse and the individual cells that were effectively the cheating cells, or these non-sticky cells, part of the mat, also collapse into the broth. Well, let's consider collapsing the bounds of this microcosm. So you could think about the glass bounds here as being like the skin of a multicellular organism. So to what extent might we think about this as a multicellular organism in the absence of the glass vial? Well, here's a possibility. So consider a pond in which are reeds. These reeds grow from the bottom and they extend out from the surface. Now these reeds provide as a focal point around which a wrinkly spreader mat, or the sticky mats, can form. These mats are not buoyant. I didn't make that point. They can't just get bigger and bigger. Their capacity to exist at all depends on, in the glass vial, sticking to the glass vial wall. In the absence of a glass vial or a very large diameter, they will not go anywhere at all. But if you put a reed in the middle of a large glass vesicle, then you will get a mat around the reed. So here we have a pond and many reeds within it. So consider this possibility. So here is, I think there's a backdrop in blue, but yeah, it's so faded, or at least the light is too large. Anyway, there is a background here of water, imagine that, and we've got some reeds, 4 reeds here extending through the surface of the pond. Let's consider the possibility that at least one of these non-sticky types survives. Remember, there's not much oxygen in here, so there's huge selection to grow at the air-liquid interface. And one of them happens to give rise to a wrinkly spreader cell, which, should it happen to find itself attached to a reed, can give rise to a mat. And one could imagine that process going on, such that these reeds become colonized by mats. Now, these mats differ in their physical properties. Again, something I didn't say earlier on, I mentioned many different genetic routes to wrinkly spreader. There is abundant variation, a huge amount of phenotypic differences due to these individual mutations. So in terms of those Darwinian properties, variation is a done deal. There's plenty of it. They differ in their rate of growth, their strength, and also the rate at which these non-sticky types emerge. So here we have non-sticky types, so-called cheating cells that emerge in 3. This one happens to get a few more, and up here we get a lot more. This one down here happens to collapse early, perhaps because the cheating cell arose too early. It had a high rate of cheat production. One way or another, the mat has collapsed, and this cell here has not enjoyed the benefit of growing at the air-liquid interface for long, and in fact you might think of this group leaving just one cheating cell. This one, on the other hand, collapses later on, and into the broth phase, into the pond, go many individual cheating cells, each of which stands a chance of acting as a seed for the formation of a new collective. So what I've shown you here is that that collective that was once here has now doubled. There are now 2 copies of it. It's emerged thanks to, or it's reproduced if you like, thanks to these asocial types, these non-sticky cells that acted as germline cells. In the meantime, this one here has been driven extinct. So we have, in effect, a process of evolution by natural selection, or the possibility of the process of evolution by natural selection proceeding here as a consequence of groups competing with groups via a life cycle that involves these cheating types that act as a kind of a germline. That at least is the idea that I'm going to try and present here, and which has motivated the experiment that I'll talk about. So here we are again, that cartoon, simple undifferentiated groups. These cheats that we see as the cell type that absolutely needs to be stopped at all cost. Well, the possibility here is that maybe the best way to beat him is to join him, is to bring this cheating type into the developmental program effectively, where it becomes part of a life cycle facilitating the reproduction of collectives. So let's look at that and see how we might progress. Now, already the sense of a life cycle here, initially there is no chance that this life cycle emerges by development, and life cycles in extant organisms are inevitably driven or under developmental control. The idea here is that, well, the reality is that each stage of the life cycle requires a mutation. So a mutation to make the mat, a mutation to make the germline. So to what extent can mutation drive a life cycle? It certainly can't do it in perpetuity, but possibly it might be sufficient to allow the possibility for selection to work on effectively a developmental program, the capacity to go through the phases of the life cycle to the point where developmental control might in fact even emerge. So to give you a sense of the possibility here, let me take you through a little bit of the genetics. So what you've got here is the outer membrane of a bacterial cell, its inner membrane, and hooked into the membrane are 3 different signal transduction pathways: the WISP pathway, the AWS pathway, the MWS pathway. Each has a diguanylate cyclase, which produces cyclic di-GMP upon a mutation which constitutively activates the diguanylate cyclase. And when cyclic di-GMP is overproduced, glues are overproduced, and the switching on and switching off of these glues allows a life cycle to proceed. So here is one example of a life cycle that runs through 8 mutations before we'll go somewhere else. Let's look at how it proceeds. So the first mutation here is in the diguanylate cyclase. That's actually a gain of function mutation in this particular instance, and it gives us the first sticky type. The next mutation, to go back the other way, knocks out the signaling or the membrane component of this MWS. Signaling is blocked, and we revert to the non-sticky smooth type. This pathway is being used, abused perhaps, and that's the end of it. Selection moves on to another pathway. Mutation in a negative regulator activates the diguanylate cyclase, and we see the next sticky manifestation. Next mutation destroys the diguanylate cyclase, and we get a smooth. So we've got a sense already this life cycle has gone through 2.5 transitions. And it can go on. It now moves to the WISP operon. A mutation in the negative regulator activates WISP-R, which becomes constitutive, And that comes down to remind me and you that we'll come back to WISPR because it plays an important role in the emergence of a genetic switch that underpins the developmental program, but we'll get to that. That causes the next wrinkly spreader form, and that process goes on. It's shut down with a mutation here, although not one that abolishes the glue, just the positioning of the enzymes inside of the cell. Interestingly, here, the next step is for evolution to go back and reuse this pathway that you might have thought would be not usable again. But it's usable. This is the negative regulator. The digonylate cyclase was never inactivated. This was a gain of function mutation. And once again, we get a transition to the wrinkly spreader mat-forming type. That process can, in principle, continue to go on. So a life cycle by mutation is not viable in the long term, but perhaps it's sufficient to get things going. So a story now, back to these experiments of two life cycles. One in which we embrace cheats and the other which we purge cheats. Actually, it's often simpler to break it down to this, but let me just take you here as a single mat-forming cell. The mat forms, we see the inevitable rise of the non-sticky types. The cheat embracing regime then gets rid of the cooperating collective, takes the cheat, and that then has to reform the mat once again. So a life cycle of two phases, cooperating cell, the mat forming cells, the non-sticky cell. Back to mat, and around it goes. The cheat purging regime, on the other hand, is the equivalent of fragmentation, but through a single cell bottleneck. So it's exactly the same in all regards, but it doesn't have a two-phase life cycle. So there is the single mat forming cell that produces a mat. We see the inevitable emergence of the non-sticky cell types, but they are now eliminated by experimental manipulation. The cycle goes through a single cell bottleneck, but it's of the same sticky cell type and back around. So here we are. The mat forms through a single cell phase, but it's more of the same. The mat regrows, single cells regrows here, the mat grows, but it then is punctuated. It must go through this different phenotypic state before it can recreate the mat again. So first of all, the cheat embracing regime, where this we have a two phenotypes life cycle. The life cycle here doesn't play out in a pond as much as I would have liked that. We did explore those sorts of experiments to begin with. The secret truth for why we didn't proceed is that once you grow these cells in large volumes, the smell is just so bad. It is impossible to even enter the lab. Anyway, here's the life cycle. A two-phase life cycle. Phase 1, which lasts 6 days. Phase 2, that lasts 3 days. A single cell, mat, forming cell, is introduced into the static microcosm, forms a mat. At the end of the 6-day period, the organism is deemed old enough, and it is harvested, and we collect the reproductive output, the cheating cells, the smooth types. The smooth types are then mixed together and introduced into a different microcosm, but also non-shaken, where during a 3-day period they must compete to be the first to give rise to the mat-forming cells again. A single mat-forming colony of the most common type is then taken around, and that refounds the next generation. So the life cycle here operates over a 9-day cycle. It's not easy for this cycle to run, again requiring not only mutation, some complex evolutionary ecology en route, but also, of course, the capacity to form the next stage of the life cycle. So these life cycles can fail, and when they fail, the organism in the tube goes extinct. It can fail if the mat is not evident at the end of the 6-day period. So we would simply consider that the equivalent of the organism having died before it reproduces. The life cycle will not complete if at the end of the mat forming stage there are no non-sticky types. So if there are no smooth non-sticky types, again there's extinction. Similarly, if at the end of the 3-day phase, the next phase of the life cycle, the mat is not being recreated, so we just have smooth types, further extinction. So extinction is guaranteed. Now, it's interesting, although extinction, death, typically seen as problematic, death as becomes possible via the requirement for these cells to run through a life cycle, ensures the possibility that selection can operate over both short and long time scales. So individual cells are doubling every hour or so. But selection also gets to work, or has the possibility to work, over a 9-day period as well. And this is vitally important. It will work over the 9-day life period, provided there is enough variation between collectives, and you'll see this in a moment. So selection is possible at 2 different time scales. Selection over the short time scale, that of the individual cell, favors rapid growth, but persistence over the long time requires cells that can transition through this cycle. So cells that grow rapidly are likely to kill the collective, and selection will eliminate that, and those collectives stand to be replaced by those lineages where the cells favor persistence of the collective over this longer 9-day time scale. As I've already indicated, this life cycle is clunky, and persistence is less than assured. So here is, if you like, the dynamics of extinction, starting with 120 microcosms that run through 7 generations. By the end of the 6th generation, all microcosms have gone extinct. So the life cycle has failed in all 120 microcosms. So, I mean, that's the end of it on one hand, and on the other, perhaps it's not surprising given what we are requiring of the cells, but there is one thing that is surprising, and that is that some lines actually do get to run 6 generations. Now, in this experiment, we've not actually allowed a birth-death process to operate at the level of these evolving lineages. There has only been death. We've not allowed the possibility that a viable lineage might export its success to replace, if you like, the body of a dead individual. And that possibility that we actually operate a birth process on top of death would allow competition to work and selection to work very potently between collectives, group selection, where the groups are Darwinian by virtue of the experimental design. If we were to allow the possibility of persisting groups to export the success, we might allow the possibility that life cycle disrupting mutations were outrun by life cycle facilitating mutations. So we allowed this possibility, selection among collectives. Here's how it worked. First of all, there's 120 microcosms. For this experiment, we broke them into 15 replicate populations. Here is one of them. Each replicate population is comprised of 8 microcosms. Here is a scheme that shows how this birth-death process operates at the level of one of these 15 replicates. So 8 microcosms. You can see some red arrows. This one is because the matters collapsed before day 6. So this lineage is extinct. And in the previous experiment, that was it. That meant that we were down to 119 microcosms and so on. Now, though, what we allow is the possibility of a line that has got through that phase of the life cycle to export its success, to effectively reproduce. It splits, and it occupies 2 places. There's the possibility here that that is persisting could go on and discover some more beneficial mutations. On average, there were about 50% of the microcosms would go extinct each generation, so 50% replacement each time. So this is between group competition that's implemented here, but there's never any mixing. That's another story. If you mix, you get a very different outcome. No mixing here. Anyway, the upshot is, whereas we saw extinction of all lines by the end of generation 6 in the previous experiment, where there is no birth process possible, no reproduction, we now see these lines run 10 generations, and they're all fine. So selection now solves the problem, or I should say the possibility of selection between collectives solves the problem of these life cycle disrupting mutations. We end up with lines that are fully viable at the end of this period. And again, as I've emphasized, death is an important part. It allows selection to work over this longer time scale. Now, for much of the rest of what follows, I'm going to give you just a vignette of what we found. Going through the data will simply take too long, but of course I'm very happy to discuss this in much more detail. So what we've done next, of course, is to ask, given that evolution on collectives has been able to operate with selection over the course of 10 life cycle generations, has selection really seen the collectives, has collective fitness increased over that period of time? Do collectives, after 10 generations, beget more collective offspring relative to the collectives at the start of the experiment? So we measure fitness of derived collectives and compete them compared to ancestral types, and this is performed in exactly the same procedure early on, but with a mixing regime. So we pit the derived and ancestral directly against one another. We have neutral markers, and here is 8 microcosms after a period of this competition, and we see there are 6 of the 8 microcosms are occupied by this type, only 2 of this type. This has left more offspring than we would expect. If they were equal fitness, we would expect 4 of this color, 4 of this color. The derived one leaves more mat babies, effectively. It is the more fit type, and we measure fitness at the level of collectives based on the number of mat babies that are produced. We are also, of course, interested in how selection sees the individual cells that makes up the collective. And we achieve this in various ways. We measure population density, growth rate, the likelihood of producing the next stage in the life cycle, and many other measures, in fact, of single-cell performance. Effectively, the life history traits right throughout these experiments for all the lines. So what do we find? Whoops. Go back, I'm sorry. Here's what we found. And it was surprising. Selection certainly sees the lines, if you like, the nascent organisms, but it sees the cells very differently. So at the end of 10 life cycle generations, what we see is that the fitness of the lines, the lineages, has gone up significantly. If it had not changed, it would be at the 1 point. That is perhaps not so surprising because these collectives are endowed with Darwinian properties by virtue of the design of our experiment, the ecology. What is surprising, though, is that the fitness of the cells after 10 generations, they've experienced around 2,000 generations of evolution, single cell level, their fitness has declined significantly. This very clearly says that we can no longer explain the fitness of collectives based upon our understanding of the fitness of individual cells. This is thought to be, well, this is often referred to, theoretically, as being referred to as decoupling, and a sense that this ought to happen during transitions in individuality when selection moves from cells to collectives. The fitness of collectives should at some point or another become independent and no longer simply the sum or the product of the cells that comprise those collectives. In this particular instance here, it's reasonable to look at the collective as a new kind of individual that is no longer simply the product of the fitnesses of the individual cells here. So what is the basis of fitness improvement? How do we get collectives that are more fit and yet the individual cells grow more slowly and fail to reach such a high population density? So what is the phenotypic basis of lineage improvement? Well, again, in cartoon form. Here is our ancestral type, dotted lines, thin arrows, indicating that the life cycle to begin with is clunky and it's unreliable. At the end of 10 generations through this life cycle regime, the life cycle for the best lines is far more reliable, full stop, it's reliable. Moreover, and difficult to see in this light, striking to my eye at least, having spent decades looking at wrinkly spreader colonies, is that in the best lines, each manifestation of the wrinkled type after each cycle, 1, 2, 3, 4 shown here, they're exactly the same. And this, at least on the basis of phenotype, the reliability and the identity of the recurrence of the wrinkly type each time smells like some sort of genetic switch, some sort of developmental program, which of course would be wonderful had it emerged. So it smells like a developmental switch in the sense of the reliability and the reproducibility of the phenotype. Well, in principle, it should be possible to get some indication of this by sequencing alone. I mean, sequencing is not genetics, but we might have got it at least at some hint of what's going on. This is the idea. So here's our experiment. Here is our ancestral type. It goes through 10 generations. We did sequence all lines, 15, but I'll just tell you about one. So to sequence at the end of 10 generations. Now the idea here is that the life cycle was driven by a mutation, but what we see here is indicative of some sort of developmental switch, which could be genetic or epigenetic. And irrespective, what we would expect if we've got a genetic switch or an epigenetic switch, certainly an epigenetic switch, certainly likely if we've got an epigenetic switch, is that if we then ran through 3 more cycles, these are expedited cycles, as is shown here, so we're going very quickly, 1 day in the matte form, 1 day in the smooth, 1 day in the matte smooth, 1 day. Then, if they've got if something interesting has evolved here, running it another few cycles and resequencing, we should see no change in the DNA sequence. If it was a genetic switch, we would think about sort of flipping backward and forward between states. So as long as we sequence the wrinkled type here and the wrinkled type here, it would be in the same state. Or if it's epigenetic, of course, we would see no mutation at all. So that was the idea. Evidence of mutation here. Something's happened here. If it's true, there shouldn't be any more genetic change. Well, we sequenced in this line at this point here and found 53 mutations, which is a lot more than we would expect. At least 1 mutation for each time, but 53 is a lot. And among those 53, we found this mutation. Now, a mutation here in mutes confers a mutator phenotype. The global mutation rate goes up about 100-fold. So this makes this experiment here now problematic. Even if there are no mutations underpinning the cycle, we will pick up mutations due to this locus here. So, of course, the obvious experiment now is to replace this mutator locus with the wild type and repeat the experiment. I fully expected when we replaced this and made it wild type not to affect this remarkable capacity and the reliability to go back and forward, but I was quite wrong. And it was, I found it really quite incredulous. What we did find upon reversion of this locus to wild type is the capacity to reproduce at this very high rate was lost. It didn't go back to the ancestral, but it certainly wasn't as spectacular as we saw it here. So that certainly says that this capacity to transition between phases is mutational, and the epigenetic route is out the door. It seems to need this mates mutation, but it seemed improbable to me, given the phenotypic identity between recurrences of the mat-forming type, that transitioning was simply a consequence of a globally elevated mutation rate. The phenotype was simply too reliable. And what about the possibility of a genetic switch that uses mates? Well, that seemed a long shot, but it turns out to be the case. So here is a slightly more elaborate form of the experiment I mentioned a moment ago involving sequencing. Our ancestral type, we sequenced halfway through the 10-generation phase. We give a slightly different numbering here, but this is 10 generations. So sequence, we expect to find mutations. We do. We find 5. We expect 7. Through here, as you saw before, we found 53. One would expect about 21, so many more, but we know that's due to the mutator phenotype. Here's our expedited 3 rapid life cycles, and what we did was to sequence the very last wrinkly and its immediate predecessor, the smooth type. They differed by just 24 hours. And when we sequenced this, we found that these two differed by the number of Gs and attractive Gs within WISPR, one of these diguanylate cyclases. We also found, when looking at the data, and something that Craig McLean has very recently reported from a mutation accumulation experiment done in a mates lineage of a closely related bacterium, and that is that the mutator, the mates mutation, not only increases the mutation rate about 100-fold, but it specifically elevates frameshift, the likelihood of finding frameshift mutations solely in tracts of Gs. In Craig's experiment, he found 10% of the mutations due to mates were frameshifts, something like 105 of them, and 101 of them were specifically pluses and minuses in tracts of Gs exclusively. So very curious. Now, what seems to have happened here is, and I'll show you a little more in a moment, the emergence of a genetic switch which involves backward and forward between 7 and 8, but it needs the mates mutation. So here's the evidence of the switch going backward and forward. 4, not 8 as we have here, just 4 replicate lines from generation 10 with the mates mutation, mates wild type over here. Wrinkly, smooth, wrinkly, smooth, wrinkly, smooth, wrinkly, or matte, non-matte. 24 hours between each cycle here. This can't do it in 24, 48 is the best we can do. The red arrows, well, in the data, the body of the table, we've either got 7 Gs or 8 Gs, and where there is a switch from 7G wrinkled to 8G smooth, whoops, we have a red arrow. So gain of G, loss of G, gain of G, loss of G, gain of G, loss of G. It's not perfect, it doesn't happen in every instance, but you wouldn't expect it to be perfect. When we see wild type, we see just one instance of a transition here. But already this capacity is very much dependent upon evolutionary history that I will take you through very briefly in a moment. Many extinction events here, so when it finds a way of going via another route, it's likely that that ends the lineage. Here, many of the changes are via a frame shift in attractive Gs, effectively a contingency locus, a genetic locus that facilitates high-frequency reversals, but dependent upon this Massey mutation. Well, also I should point out, although this mutation, of course, arises originally in a single cell, and only one lineage of out of the 120 ever evolved this, for reasons you'll see in a moment, it subsequently spread. It rose about generation 7. And over the course of the rest of the experiment, it not only swept to become, to fix across all microcosms in one replicate, there was a wholesale extinction of another rack. And it, line 17 here, also founded this other microcosm. So in fact, it had spread very quickly across 2 replicates in this experiment. So the mutation evolved in a single cell, but that mutation greatly facilitated persistence of this lineage through the life cycle. In fact, it clearly facilitates the life cycle. The likelihood that this evolved is to me quite remarkable, and it really is an instance of remarkable chance, but also some important history. So here is a cartoon of the genome. These are diguanylate cyclases, or operons containing diguanylate cyclases. There are 39 diguanylate cyclases in the genome, so the potential for a lot of evolutionary innovation here. Here is the non-sticky form. First mutation in one of these loci gives us the wrinkled sticky matte form. Next mutation shuts it down. You sort of saw this earlier on. That is now used, it's out of the way. The next mutation that activates the mat is in another locus, and again it shuts down to the smooth with a mutation in that same locus. That's now dispensed with. We see the WISP operon with WISP-R being used early on. WISP-R becomes phosphorylated, and here's the 7G residues that span, in fact, the active site, GGEEF is the active site of the enzyme. The next mutation, though, has nothing to do with the WISP-R gene itself, but elsewhere in the operon, and it shuts that down. Evolution goes and works with another locus. We see the same deal going back, and eventually it comes back to this operon. In fact, it's done it 5 times. Now, the chances that it leaves this operon intact so that WISP-R is constitutive is extremely small, but it does. And what then happens, the next mutation is MUT-S. MUT-S now, given that these other pathways have largely been eliminated, these are easily trod pathways, we now see this 7 base pair sequence with significance that it previously didn't have. It now becomes highly likely that the switch off and back to the smooth takes place as a consequence of a single G. Or loss, we have some instances, not in the experiment I just described, where we get going from 7 to 6. We get a frame shift. That destroys the reading frame right in the active site, and it reverts the phenotype. And that stands to switch, toggle backward and forward, as I showed you just a moment ago. It's reasonable to wonder that the possibility that this is very much a dangerous liaison. We have a genome here with an elevated mutation rate, but it's also capable of doing some remarkable behavior. Effectively, this mutation has brought about 2 different phenotypes. They're different aspects now of a single organism. Nonetheless, we have the likelihood of we have a deleterious load due to the fact that most mutations are deleterious. However, just some back of the envelope calculations here, in context of the ecology of the organism here, draw attention to the enormous opportunity for purifying selection to get rid of those deleterious mutations. A single cell each time passes through a bottleneck as you transition. Anything that's loaded up with deleterious mutations is not going to make it through that transition. The worst case would be make it through the transition, but there is a mutation that destroys the wrinkly on next manifestation. But provided you've got the process of lineage selection going on here, then that doesn't matter. It will simply be replaced by another type that has not got that deleterious mutation. So it's quite possible that, at least for a while, this is a good liaison, that the costs of, the normal costs of an elevated mutation rate are eliminated due to, thanks to, the possibility for purifying selection that exists within this life cycle. Okay, we're just about there. The causative factor. So I've put a lot of emphasis on this life cycle with two different phases. What about fragmentation, this control experiment where we take the same life cycle, but now we don't embrace this alternate state. We simply have a one-phase life cycle, which involves a large mat and then through a single cell. Large mat, single cell. It's fragmentation, but through a single cell bottleneck. Well, exactly the same process. We allow selection to work between the groups. Again, identical in every way, but not through the second phase. You saw a figure like this before. This is the outcome of 10 generations of selection at the level of life cycles, at the level of the mats over this 9-day phase. And the fitness of collectives has gone up, and we would expect that, given the opportunity for selection to work on collectives by virtue of the ecology that these organisms experience. But here we see that the fitness of the individual cells that comprises the mats has also gone up. So nothing particularly surprising in that. One would expect this and might have expected it for our other experiment as well. Selection here has, or one way of thinking about this is that the fitness of collectives is explained readily by the improved fitness of the individual cells that make up the collectives. There's certainly no sense of decoupling here. So it would seem that this life cycle of two different phases does offer to selection something rather interesting. So a couple of slides by way of perspective, and this could easily spill over into discussion and beer, so I will perhaps keep it short. I've talked too long anyway. So back here, the origins of multicellularity in this simple cartoon. Individual cells, collectives, the problem of cheating types, the emergence of Darwinian characteristics that allow these collectives then to participate in evolution by natural selection in their own right. Complex biological life at the end of this seems more or less inevitable. Critical here is this capacity for collective level reproduction. Without collective level reproduction, we're dealing with an evolutionary dead-end situation. There is no possibility of selection working on collectives in their own right. And I've already emphasized that fragmentation is not the only way by which nascent multicellular collectives or which reproduction might emerge amongst nascent multicellular collectives. In fact, my colleague Eric, who's now at Santa Fe, we explored alternate routes by which the earliest groups might reproduce. Fragmentation is one. The life cycle that I've described here is a second, and there is another similar configuration that could also emerge very early, which involves two distinct cell types. More in there, or we could talk about it later. But here what we've seen is that capacity for reproduction via this alternate and seemingly complex cycle can emerge actually relatively easy. The critical or the limiting factor really are the ecological circumstances that would ensure that this life cycle would play out. The life cycles themselves are very interesting, and their origins are not much thought about. I've got very interesting, indeed, some work with Sylvia de Monte, who's also here, has got us even more curious about life cycles and the possibilities that they provide for evolution to work. It seems that once you've got a life cycle with at least two different phases, for that life cycle to work, it's not possible for selection just to work on this state or this state. It needs both. Otherwise, there's extinction. Once you've got a cycle like this, selection sees effectively a developmental program. It gets to work with something altogether different. Whereas, if you have a life cycle that's via fragmentation, really it's just growth through a bottleneck, growth. Selection doesn't get to move to any other property other than that of the individual cooperating cells. One might look at this and say that traits that yield success at the level of collectives are simply those that determine success at the lower cell level. And that is definitely not the case up here. In fact, you end up with a situation, and it's early days in these experiments, but we already see with this Mates-dependent switch, we have effectively these two different types of now different manifestations of a single organism whose evolution is unified via a developmental program. I'm going to skip over this. Here I would make an argument that, or for a perspective, that is distinctly multi-level. We could talk about that. But it would take too long to explain at this particular point in time. Back to this comment I made a number of times about levels of selection, but also the experimental design. And here, I would say many different perspectives are possible when we think about evolutionary transitions, and we necessarily get into rather uncomfortable territory as we go from thinking about selection working on individual cells to it beginning perhaps working on collectives of cells. Once we've got full-blown multicellular organisms, it's no problem. But as we go through these early stages, there inevitably is some point at which selection begins or suddenly moves. So the tempo, the dynamic of how selection moves is something we really don't know too much about. And again, one could offer a variety of different perspectives on this. There is, in our experimental design, again, an ecology that is distinctly multi-level. The two different timescales, in fact, define these two different levels, that of the individual 1 hour replicating cell and that of the 9-day period. And that means that selection can work over these two different timescales. Selection on cells is favored by the short-term interests of selection, but such short-term growth is unlikely to promote persistence over the long term, over the lifetime of this cycle. And again, persistence in this particular instance in the life cycle requires more than just switching between sticky cells and non-sticky cells. It requires this developmental program that underpins expression of this mat forming phase that effectively does two things. It must do two things. There is the need to build the body of the organism, which is important in terms of acquiring a limiting resource, oxygen. But at the same time, those cells must produce the seeds, effectively, of the next generation of mats. What we really have here is a reproductive division of labor. And that very much fits our understanding of that with a multi-level selection perspective. Just one final word on the decoupling here. What we would expect, given that there is the possibility, thanks to this life cycle and its fallibility, the possibility that selection operates over the long time period means that selection can, provided there is enough variation among collectives, selection over the long time scale can conquer selection working at the level of the individual cells. And when that happens, and the thought is that theoretical expectations is that should happen and has happened during evolutionary transitions, we should see the lower level entities effectively beginning to work for the good of the collective, or another way, to have been de-Darwinized by selection operating over this longer time frame. So thank you. Thanks again to Katrin and Caroline, who have done a heroic amount of work. Eric Libby, who was our resident math guy, Yuri Pishigan, a PhD in math. Yuri's now at Santa Fe. Ben Kerr at the University of Washington. Sylvia De Monte at École Normale, who's also here. And a special thanks to Jonas Korlak at PacBio, who really helped us out when we needed to sequence an inordinate amount. And the PacBio technology has been a very interesting experience for us. If you're interested in knowing more about that, I'm around for another couple of weeks. Thank you. Questions? Pleasure. Yeah.
Well, first, thanks for a very interesting talk. I was wondering a bit, like, I can imagine that there will be competition between the two-stage life cycle and the one-stage life cycle, because you treated them as separate, but they will co-occur, or they may co-occur.
That's right.
At least. Have you any thoughts?
Yeah, well, co-occur, yes. I mean, what we do by having the body around, so the emergence of bodies and boundaries is really important. Integration. We have no problem looking at you or I and saying we're multicellular because there are boundaries. There are many things that don't have such distinct boundaries. Now, one thing that boundaries solves is heritability. If things can mix, heritability becomes problematic. And if we don't have heritable variance in fitness, if there's a lot of mixing each time, then selection won't work on collectives. So boundaries are important. So if you had complete mixing and the two strategies competing in an ecology that didn't prevent mixing, I think it would be a mess, and probably heritability would be low, and the opportunity for selection to work with sort of powerful handles would be denied. That said, I think that's true. If there's mixing, nothing happens. If there was limited mixing and you had the two strategies competing, I'm pretty sure, my intuition tells me, that selection sees something altogether different when it sees this life cycle of two phases. You really have entities that are reproducing. Effectively, it delivers to selection a machine that it can work with in a very potent way. So, provided you didn't have complete mixing each time, then I think away it would go very quickly. Selection would have a machine to work with that is very potent. You have this sort of feedback between form, function, and what selection sees. Of course, there's no sense that it needs to be like that, but I think when these configurations emerge, I think they are seen by selection in a very potent way, and selection just doesn't see it, I don't think. But a model here would be very interesting, actually. We could talk on, this is something we've very much talked about, actually. Competition between different configurations and whether, in fact, some would really take off by virtue of some, as I'm indicating, properties that are handles, effectively, that are delivered to selection.
Question at the back, maybe?
Yeah, sorry, go ahead.
We don't have this. We have another one here. Matt?
So suggesting we show mutations driving the life cycle.
Yeah.
So, less interesting question is, wouldn't you therefore predict that population size would determine how that would happen, how quickly that would happen? But then,
right.
secondarily, are you extending that to suggest that this would explain many instances of the evolution of?
No, well, I guess a number of things to throw in there. Of course, life cycles can't be driven by mutation. Well, they're not, However, a genetic switch is a long way down the path to development. One could easily imagine this becoming far more refined. I mean, this is only some 10 generations of life cycle evolution. But already what it has done, of course, is effectively, so one of the problems that emerges early on for these organisms, again, it's both a blessing and a curse, is that each next manifestation, heredity is slightly reduced because the mutation that gives the first wrinkly is different to the one that makes the next smooth, which is different than the one that makes the next wrinkly. So you've got certain imperfection in the life cycle, a lot of clunkiness, and heritability is not great, although probably the rate, we've argued elsewhere, is heritable. But once you end up with a genetic switch, I mean, you have got one organism that expresses two different phenotypic states. And there are, plenty of examples out there there are examples of differentiation due to switches, genetic switches, epigenetic switches. So it's sort of a hop, skip, and a jump. It's not very elegant, it's not refined, but one could certainly imagine it being worked upon and being refined far more elegantly. The other thing that's kind of curious that your comment makes me come back to is, when the life cycle is being driven by spontaneous mutation, you have a lot of variation that is effectively being presented. Each time you've got a different variant of a wrinkly, a different variant of the smooth, different evolutionary history. There's a lot of potential there, even though we only have 120 microcosms. Now, once you get to a genetic switch, you no longer have that, or developmental control. Each recurrence is effectively, barring the odd mutation, exactly the same. And I've almost wondered if this might not have made sex particularly beneficial at this early stage. But this mutator in this background here, although you're going now reliably through the cycle, the mutator is also generating variation at a much higher rate, and maybe that turns out to be very important in the long run. So the short of it is, I think it's clunky, it's early days, but there's the potential there for it to be much more refined, and it certainly has in that one line that we've looked at. That capacity to switch dependent upon mates means that you have a single organism that is switching back and forward between two different phenotypic states, one with a soma-like function, one with a germ-line function, in a reliable manner.
One last question.
Yeah.
I'm probably really slow. Could you go through it again and explain why the two stages is going to give you the integration more? I mean, theoretically, in models, it's it's been reasonably straightforward to get group selection to operate without that two-stage life cycle. So what is it that makes it special?
Well, first of all, the standard groups, trait group models, and so on certainly show that selection can work on groups, if that's what you're thinking. But
but even without fragmentation, it works.
but there is an assumption. I mean, those models typically do not consider reproduction of groups. In fact, their purpose is to explain how cooperation can be maintained. So what this does, and really those sorts of trait group models, for example, there is no heritability there. I would say there's almost no potency for selection to work at the level of collectives. So what we've done here, the way of thinking about our groups is very much as Darwinian entities. These are groups that beget groups, and we ensure this by virtue of the ecology. What is it about the two-phase life cycle? Again, I think it's that selection, let's say with fragmentation, selection simply sees a cooperating cell. That's a trait of an individual cell that has relevance in the context of the collective of cells that does this. Selection sees that. That's all selection gets to work with. In fact, how you even get fragmentation evolving in a sophisticated way without us interfering to do this is far from clear. But basically, selection just sees more of the same. Whereas when you've got a two-phase life cycle, selection, where both are ecologically relevant and part of a cycle, then selection cannot just see one type alone. It needs to see, if the cycle is going to run into perpetuity, it needs to see effectively a developmental program. And that developmental program is an altogether new thing. And I think that's, effectively selection has delivered something altogether new that is no longer just the product of a single cell.
There's a temporal component to it.
There's certainly a temporal component, spatial as well, but certainly a temporal component, yeah. And the other thing that a life cycle does with this fallibility, I think it almost sets this, well it does, it sets, I mean we set a 9-day life cycle, but if you had a life cycle, it sets the longer time scale. And once you've got selection working over a longer time scale, and it's very marked once you've got a two-phase life cycle, then you really, I think, have possibility for selection to work potently at the level of that longer time scale. If you don't have a second phase and it's just fragmentation, selection just, again, sees more of the same. Even if you go from a single cell to a large group, a single cell to large, it's growth, really, if you like. It's growth rather than reproduction. But the main thing is, I think, the cycle, 2 phases or more, selection has to work on a developmental program to make that cycle work. Otherwise, I mean, one possibility just goes extinct. But if it can work, it sees something different. Possible?
Well, we'll discuss afterwards.
Yeah. Thank you.
I think everybody's ready for drinks, maybe, but if I may throw one last one in.
Yeah.
So you presented this as a life cycle selection, but what do you think about environmental fluctuations? Because there's a good body of theory which suggests that environmental fluctuations can drive evolution of phenotypic variability, which I think you refer to as genetic switches.
Yeah.
So then, it could be generalized because there could be many different environments that the organism experiences, and many of them might fluctuate.
Yeah.
So in that sense, do you see then that all the phenotypic variabilities that can be there as a manifestation of multicellularity?
Well, no. Again, I would, you could define multicellularity in many different ways. My stance is to sort of look at the collective that you want to tell me is multicellular, and I would ask to myself, to what extent does that collective, and it could be very loose and not terribly well indicated, but to what extent does that collective participate in the process of evolution by natural selection? So to what extent does it manifest some semblance of heritable variance and fitness at the level of collectives? Now that could be brought about by very ephemeral circumstances, changes in the environment that might create a collective phase and then a dispersal phase. That's possible. But I think one of the challenges that seems to have been solved by organisms is to endogenize these cycles. And if there is complete reliance on the external environment, there's probably limited, depending upon how reproducible. Whereas, I didn't emphasize it here, but one thing that drives the life cycle, the 2-phase life cycle, is the niche constructing activities of each phase. It effectively changes, each phase changes the environment and it sets the stage for the next phase of the cycle. In fact, Eric and I had something out, +1 last year, where we argued that these sort of cycles facilitate endogenization of signals, which we argue could be very important for the emergence of development. So you need some sort of reproducibility, some regular cycle. Now there are possibilities, and we see lunar cycles, day-night cycles. These are possible. Whether the extent to which they played a role in the emergence of multicellularity, I'm not sure. The sense is the problem is how you get something that is reproducible with the external environment that is also creating collectives and leading to their reproduction. I don't know. We can talk much more about that, I think.
All right. If there are no other questions, we can take this to the wine and continue it over wine. And let's
Thank you.
all again.
Thank you. Thank you. Thank you.